So in this lesson, we're going to continue to explain the roles of plasmid in the transfer of the gene into an organism. Describe and explain the steps involved in the polymerase chain reaction, which is in short PCR, to clone and amplify DNA, including the role of PEG polymerase. So what is this? I'm going to explain this one later on. And lastly, we're also going to look at the promoter, right? Why promoter may have to be transferred into an organism as well as the desired gene. It means when you transfer the gene, you also must transfer promoter together with the gene one. So these are the 3 learning outcomes in the syllabus that we're going to cover in this lesson today. So let's talk about gene delivery. Gene delivery methods by the vectors. So there are many different types of vectors that we can use to deliver the gene to the recipient organism or recipient cell, recipient cell. So we can use plasmid, right, to deliver a gene to a bacteria. So this is a plasmid here. So we can put whatever gene that we want to deliver. We cut and paste this gene to this plasmid and we transfer this plasmid to the bacteria. Okay. We can use bacteria as a vector to transfer the gene into plant cell. So you look at the second diagram here. Second diagram here, can see, okay. So this is a plant cell and this is a bacteria, and this bacteria got plasmid in it. And then this plasmid contains the gene that we want to deliver to the plant cell. So we infect the plant cells with the bacteria. So the bacteria will now transfer its genetic material, the plasmid, into the plant cell. So the gene is transferred to the plant cell. So bacteria act as a vector to transfer the gene. Next, we got gene gun method. Gene gun method, you can see here, this one, it looks like a gun, right? So when you have a gun, you must have bullet, isn't it? So what they do is that, what scientists do is that they will coat the bullet with the DNA. Then they use this gene gun to produce a high pressure to shoot this bullet into the plant cell. So they need this because plant cell got cell wall. So cell wall is rigid. So they need certain pressure to actually insert the DNA through the cell wall into the plant cell. So they use a gene gun method. Then protoplast fusion method. Protoplast here is actually the part of the plant cell where you have removed the cell wall. Okay, you have removed the cell wall. So when we want to mix the DNA of 2 different plants into one cell, 2 different plants of different species into one cell, we can first remove the cell wall, and then we allow the 2 cells to fuse. So when the membrane fuses, then the DNA of these 2 cells will become present in one cell already. So this is how we transfer the DNA from one cell to another cell using protoplast fusion methods. This is only for plant cells. Then we can use virus. Now, some viruses, they can infect bacteria, some can infect plant cells, some can infect animal cells. So because naturally they can already infect the cell, we just use them to deliver the gene to the cell. But first, we must insert the gene into the virus, right? Then when the virus infects the cell, it will inject that gene together with the plasmid, whatever, so not plasmid, sorry, together with the DNA or RNA of the virus into the cell. Because virus can carry either RNA or DNA, right? Okay. Right, next, we can use liposome. Lipo means lipid. Some means body. So liposome is a structure made up of phospholipid bilayer. So this is the lipid that they mentioned here. Some means it's a structure, it's a body, it's a structure. Okay, so when we can use this one to deliver drugs, to deliver DNA, to deliver gene into animal cell. So all we need to do is that we have to allow this liposome to go near to the cell, and then the membrane of the liposome will fuse with the cell, and whatever content inside will be released into the cell. Okay, so this is liposome. So therefore, among all these vectors I mentioned here, 6 of them, there are only 3 that you need to learn in this chapter, which is plasmid, which I've highlighted already, virus, and liposome. So today we only talk about plasmid. Oh, I missed one. Micro-injection. Micro means tiny, very small, right? Injection. So this picture on the left bottom here showing micro-injection. So you got a suction pipette to hold the cell. So this cell is held by the suction pipette, and you've got a pipette with very sharp end to inject a piece of gene right to the nucleus of this cell. So it is called micro-injection. Okay, so usually we can inject this into the zygote. Okay, because from zygote, when the cell divide, all the cells will contain this gene already. Okay, that we've introduced. Okay, so let's look at the plasmid. So you know that plasmid, right, is a circular DNA that is found in bacterium, other than its original DNA, which we call them chromosomal DNA. Now, they're not chromosome, because this is the main DNA of the bacteria, so we call it chromosomal DNA, this one here. So the main DNA of bacteria is usually bigger one, so we call it chromosomal DNA, okay? But it's also circular. Now, plasmid is a small, definition, small, circular, double-stranded DNA in the bacteria, other than the main DNA, which is called a chromosome. And usually when they say a bacteria is antibiotic resistant, it's because their plasmid will be carrying antibiotic resistance genes. So usually those genes are carried in the plasmid. And then plasmid naturally can be transferred from one bacteria to the next bacteria one. And the two ways that the plasmid can be transferred between bacteria. First one is horizontal transfer, and second one is vertical transfer. So what is horizontal transfer? Horizontal transfer means transfer of the plasmid between bacteria of different species. So if you look at this picture here on the top right-hand corner, you can see that these two bacteria, the structure is different one, right? This is rod-shaped but very long, and it looks like a lot of flagella-like of structures around it, right? And then this bacteria is also rod-shaped, but it's shorter. So because of the structure is different, they're actually different bacteria. So what happens is you see there's a tiny little tube that connects between these two bacteria. So what is happening here is one of the bacteria has replicated its plasmid and then send the replicated plasmid through this tube to another bacteria. So this tube is called the sex pillars. Sex pillars, and this process is called conjugation. Okay, so second way of transferring plasmid from one bacteria to the next one is called vertical transfer. Vertical transfer means transfer from parental cell to daughter cell. When the parent cell divide from one cell to become two cells, every time when a cell divide, before that they have to replicate their DNA. So the plasmid is replicated and then it's transferred to the daughter cell. So let's say this is original bacteria, okay? So it's got this plasmid here. So from one plasmid, in order to replicate, it will, before the cell divide, the plasmid will replicate into two, then the cell will divide into two, right? So when the cell divides into two, each daughter cell will take one copy. So this is called vertical transfer, right? From parent cell to daughter cells. Okay. Now the process of bacterial division is called what? When bacteria divide from 1 to 2, this process, what is the name given for this process? Lim Yi Hao. Mitosis. Can it be mitosis? Mitosis is nuclear division. So bacteria got nucleus or not? Bacteria got nucleus or not? No. So if no nucleus, no mitosis, right? If no mitosis, then what is another word for the cell division? Very good. Binary fission, right? Okay. Binary fission means to say what? Binary means two, right? Two. Fission means to split or to break into two, right? Right. So therefore, it's split into two. So it's binary fission. So cannot mitosis, right? Okay. Now this is a common mistake among the students one, because our cell divide is definitely mitosis. But don't forget, mitosis is what? Nuclear division. Bacteria cannot mitosis on. Okay, so after saying so much about plasmid, let us look at one example of plasmid that's been used in genetic engineering to produce insulin. So this plasmid is called plasmid PBR322. It's just a name given to it like that. Your car, this is Proton Saga, this is Proton Vira, this Toyota Corolla, this Toyota what? The latest Corolla, Toyota Cross, right? The latest one, okay? So this is just a name given to the plasmid. So it was used in the production of insulin. Okay, So I will show you the plasmid later on. Let's look at why scientists choose plasmid to be used as a vector to transfer a gene to the bacteria. Okay, number one, because it is small. You know, compared to all the DNA in the bacteria, plasmid is smaller. Small means low molecular mass, so it can be easily taken up by the bacteria. Why? Because you have to pass through the cell wall and the cell membrane. So if you want to transfer from one cell to another different cell, right, it has to pass through the cell wall and pass through the membrane. The smaller, the better, the easier, right? Okay. Now, it contains an origin of replication. That means to say that when you transfer this plasmid into a recipient cell, let's say this is a recipient cell, right? You transfer this plasmid into the recipient cell. Okay. Now, you want this recipient cell to actually replicate this plasmid so you can continue to divide and form more plasmids, right? Once you introduce to one cell, you want this cell to continue to divide. That means you continue to replicate this plasmid. So every plasmid got an origin of replication. Origin of replication means what? So this is the plasmid, right? So there's one particular location inside this plasmid where it is the origin of replication, where it will start to replicate. So because it's got origin of replication, so from one plasmid, you can form two plasmid in the cell when the cell divides. Okay, it can replicate when the cells divide. So then you can clone the plasmid. Clone means make multiple copies. Identical copies. Clone. Clone means identical copies, right? To clone means you want to make identical copies by, in this case here, DNA replication. Okay. In this case here, you want to clone what? DNA. By what? DNA replication. So that's why I say this chapter is very easy. Right? Everything is basic. Everything is basic. It's just that how they use their knowledge of basic knowledge to put into a way that's useful. Okay. Next. Now, this plasmid is very interesting. We can design the plasmid to contain a few other things based on what we want to use it for. So I give you the plasmid. You can design whatever you want. That means you already got the tools to cut and paste the DNA, right? You got the tools to cut and paste the DNA. So now you can actually take it and design your plasmid in many ways that you wish, depending on what you want to use it for. So for example, the scientists can be adding several target sites in polylinker for different restriction enzyme. Now, meaning to say that the scientists can actually add in a short length of DNA into this plasmid, so insert it into this plasmid, and this short length here contain a few restriction site. So let's say this restriction site for restriction enzyme 1. This is restriction site for restriction enzyme 2. This is restriction site for restriction enzyme 3. Okay, so I add this piece of DNA, which I call it polylinker. Now, linker DNA stands for DNA that are non-coding. Non-coding means got gene or no gene. Hello? Non-coding means got gene or no gene? No gene, right? Okay, so non-coding means they don't have gene. But they've got, in this case, they've got target site for restriction enzyme. So that after I added in the polylinker, I can decide to cut this DNA using the restriction enzyme, right? If I want to cut this DNA here, I must use restriction enzyme that can cut here, right? If I want to cut the DNA here, I must use restriction enzyme that can cut here. If I want to cut it here, I must use restriction enzyme that can cut here. Right? Okay, so I add this polylinker in so that I can cut it. Now, I can also add one or more marker genes into this plasmid. So say, for example, I add a marker gene here. Okay, so this marker gene will code for something that is highly visible, so that whichever bacteria that has taken up this plasmid, let's say, for example, this is the bacteria. So I got a lot of bacteria here. I'm trying to introduce a plasmid to the bacteria, but usually only 1% of them will take up the plasmid. So among all these bacteria, say, for example, only one bacterium has taken up this plasmid, this one. Let's say this one has taken up already. The rest did not take up. So how do I know that, oh, this bacterium has taken up this plasmid here? Now, because I have inserted this additional gene into the plasmid, and this additional gene is called the marker, okay, the marker gene. So when it's inside this bacteria, when this gene is being expressed, it will produce a product that, for example, will fluoresce. Let's say you produce a protein that will fluoresce under UV. So when I shine UV light to all these bacteria here, which one will fluoresce? Only this one, right? Because only this one will produce protein that can fluoresce, right? So in that way, I know that, my plasmid has already been taken up by this bacterium. So this bacterium is a transformed bacterium. The rest are not. So this is the use of the marker gene. Okay. So do you see how wonderful it is just by using the tool of cutting and joining? And then you know what is the function of, you know what is target site. You know what enzyme can cut the target site, cut the restriction site. You know what is marker gene. You can design the plasmid and use it for other purposes, right? So this is to show you the power of applying the knowledge to design things that are useful for human. So example, gene coding for enzymes that produce fluorescence. So the marker gene I mentioned just now is actually this gene here. Okay. So why we use plasmid? Because we can insert a sample gene into this plasmid to be transferred to recipient cell. So for example, if I want to insert, say for example, Shreya, is my Shreya here? Okay, Shreya. So what gene do you want to insert to the recipient cell? Tell me. Okay. Any gene? Yes. Human gene. Because you want the bacteria to produce the product insulin. Okay, so let's say for example, thank you, Shreya. So you want to insert insulin gene into the bacteria. So it must be transported by plasmid. So therefore you have to insert first that gene into the plasmid, right? You cut the plasmid, you cut the gene and you put them together, join them, right? Cut and paste. So now this insulin gene is already located in the plasmid. So this is your design plasmid that you use as the vector to transfer this insulin gene into the recipient cell, the bacteria, and then the bacteria will divide. Never mind, forget about it, transfer into bacteria, full stop. Okay, full stop. Right. So this example of the example of plasmid that has been designed by the scientists, have been used in genetic engineering of bacteria to produce insulin. So they basically use this vector to transfer the insulin gene to the bacteria so bacteria can produce insulin. So let us look at the components inside this plasmid called PBR322. So there's an origin of replication, and then there's this polylinker which contain 3 target sites for 3 different restriction enzymes. And then you got 2 additional genes here. One they call ampicillin resistant gene, and the other one they call tetracycline resistant gene. Now these 2 genes are actually the markers. So in the past, scientists used antibiotic resistant gene as a marker to find out which bacterium has taken up this plasmid. Okay, so these 2 are actually markers. Markers means the gene that's used for you to identify the transformed bacteria. Okay, so these are the markers. Finish. Now, in the process of isolating plasmid from the bacteria, so I'm going to show you how the plasmid is being isolated. Step number 1, we have to actually hydrolyze the bacterial cell wall using enzyme. So we hydrolyze it away. And then we will then centrifuge the solution of bacteria. So when you centrifuge, right, you put the bacteria solution into a small tube, and you place a small tube into a machine, which we call them the centrifuge. So, and then after that, the machine will start to spin already. So when it spins, it will create very strong gravitational force downwards, which is many times of our gravitational force, so that the suspended particles can settle down quickly, right? So once it settled down, there'll be two layers that form. You see the top layer here and the bottom layer. So the top layer is called the supernatant. The bottom layer usually is something that is solid, is called pellet. So whatever that is heavier will settle to the bottom to form the pellet. Whatever that's lighter will be found suspended in the solution called supernatant. So over here, what we want to separate in the bacteria is the DNA from the plasmid. So when we do centrifugation, which one will settle to the bottom? Which one will settle to the bottom, the DNA or the plasmid? Which one is heavier? DNA, right? So the bacterial chromosome, the bacterial chromosome will settle to the bottom. So the plasmid will be found in the supernatant. So then when you want to use a plasmid for subsequent steps, right? For example, you want to cut the plasmid, you want to insert the gene, we will take this solution to continue with subsequent step. Okay? Right, so now we're going to look at an overview of genetic engineering, the steps involved. So just now we look at the function of the vector. So